Okay, I would like to welcome everyone to this series of lectures that represent the 10 research themes of Leiden University Medical Center, and we kickstarted with theme infection, today's subject. I'm Maria Jasdanbach, and on behalf of my colleagues, Erik Snijder, Mette Rustenberg, and also on behalf of Sander de Jong, who coordinates and drives the theme infection, I will give a short introduction. So the building blocks of theme infection are really the departments within LUMC, not only Leiden University Center of Infectious Diseases, which consists of departments that the core business is to do research in infectious diseases, but also other departments with many researchers, clinicians that work in this area. And not only do we have people in LUMC, but also we have connections with Leiden Bioscience Park and also Campus The Hague, as well as different faculties of Leiden University to work together, but also very importantly, our partners, national and international. They are important to us to form cross-sectorial partnerships because that's such an important way of approaching infectious diseases to combat them. Leiden Bioscience Park, science faculty, social science faculty, Campus The Hague, strategic European partnerships, and also strong links with institutions in Global South form the theme infection. For us, it is important that we develop knowledge-driven strategies to combat infectious diseases through development of novel vaccines, drugs, and immune modulators. We would like to be a partner for Sustainable Development Goals of United Nations by focusing on vaccine development, vaccines as cornerstone of prevention in public health, drug development for cure of emerging and re-emerging infections, and antibiotic stewardship, and highly specialized diagnostics. And all this is supposed to feed in to ensure health at the population level and also patients. And our clinics specialize in elderly, taking care of elderly, immunocompromised, migrants, travelers, and the Global South. As an example of how important it is to do integrative research, clinicians, basic scientists, and technologies, cutting-edge technologies have to come together. Here is really how we like to see vaccine development within our team. So the vaccine development research, which has very strong components such as challenge models, research into adjuvants, synthetic chemistry from the science faculty, all kinds of cutting-edge technology platforms that help us do systems immunology, multi-omics data, all these biomarkers. These strengths can be combined with the strengths in our environment at the Leiden Bioscience Park, where vaccine product development takes place. Together, it's possible to then quickly develop vaccines that can move to be tested in populations and patients in trials. And what you learn from the trials has to be fed back into this vaccine development pipeline so that vaccines are continuously improved. As an educational institute, we can also contribute towards developing the workforce that specializes in vaccinology. So our knowledge can be presented during curricular courses for our students, and also specialized, for example, courses such as summer schools or postgraduate education. With this, we really form a platform that can deal with the future infectious diseases that need vaccines to be developed rapidly and continuously. Another interesting area that is important that we would like to showcase today is really our approach to COVID-19 pandemic. So it's really been an interesting approach to bringing, again, clinicians, scientists, and technologists together and to work towards beating COVID. And with that, I'd like to give the floor to Dr. Anna Raukens, infectious disease specialist, who's been an important person in setting up Beat COVID. With this, Anna, the floor is yours.
Thank you very much, Maria, for this introduction. Indeed, I will introduce the Beat COVID consortium to you. So, of course, COVID-19 does not need any introduction, but I would still like to show you what the situation is today with about 120 million cases in the whole world. And just to take you back a little more than 1 year ago, this was February 2020, when the infection was still contained in China. We were all looking at China, and there were about 40,000 cases. And 1 year ago in March, we had the first patient diagnosed in the Netherlands. And of course, clinicians were, from that point, really challenged in how to treat these patients. So we had to, all we had was the preliminary reports from China. And from these reports, we heard that corticosteroids were not beneficial. Of course, now we know that dexamethasone has really a benefit for hospitalized patients, and we treat all our patients with this corticosteroid. IL-6 was a predictor of severe respiratory disease, and indeed now we give tocilizumab, which is an anti-IL-6 receptor antibody, as a treatment, especially for ICU patients, but also on ward patients. We thought that hydroxychloroquine was beneficial, but unfortunately this was not the case. And remdesivir, the antiviral, seemed to have potential benefit, but also now we know that it doesn't really have a benefit on severe disease or mortality. And all this was investigated in less than 1 year in global clinical trials, in which also 2 of these, the LUMC, participated. So what we had as clinicians to predict how our patients would, with COVID-19 would do at their hospitalization were these: higher age, higher body mass index. I'm sure everyone is aware of these risk factors, but we really needed more. And we needed more to make a prediction, and also a mechanistic model, also to think of new treatments which would really benefit these patients. So we brought together clinicians and researchers to try to make these models. With a lot of colleagues from the LUMC, we set up this consortium, the Beat COVID consortium. And as you can see, a lot of research groups participate, everyone very enthusiastic. And still until now, this consortium is doing this research, and we have now around 200 patients included, with multiple sample points on which all these laboratories do their investigations. So from this consortium, we have 4 excellent speakers today. Of which the first is Dr. Erik Klok from Hemostasis and Thrombosis, and he will talk about thrombotic complications in COVID-19. The second speaker is Dr. Simone Joosten from the LUCID, immunology researcher, and she will talk about the assessment of local and systemic immune responses during COVID-19 infection and disease. Our third speaker is Dr. Marjolein Kikkert, from the research virology laboratory, and she will talk to us about the virus and the vaccine. And the last is Dr. Meta Roestenberg, who will talk to us about the controlled human infection model of SARS-CoV-2. All the questions, you can write them down in the Q&A, and we will discuss them after the 4th presentation. So to start off, I would like to give the floor to Dr. Erik Klok.
All right, thank you very much, Anna. It's a pleasure to be here, invited among infectious disease specialists, and to talk about, to give you a touch of flavor of the studies that are currently being performed in the LUMC with regard to the thrombotic potential of COVID-19 infection. And I will present this on behalf of many, many researchers involved in all these studies, which I will, of course, highlight during my presentation. Let me take you back to, let's say, February last year. There were the first large reports from Wuhan, China, on the course of disease of patients admitted to their hospitals with COVID-19. And what struck immediately, at least in my, let's say, clotting eyes, was that all these reports they showed us that there was a high prevalence of clotting activation among patients with COVID-19, and especially those patients with the most severe disease. And markers of clotting activations were indeed associated with poor prognosis, as shown on this slide. Strikingly, we of course know severe infectious diseases, they may cause clotting activation. It's called diffuse intravascular coagulation, DIC, and we know this is associated with poor prognosis, but also with bleeding events and thrombotic events. And to our surprise, none of these initial reports from China reported any thrombotic complication. And this was, of course, not understandable for us. And as soon as Netherlands was hit by COVID-19 and our hospitals were filling up with patients, together with colleagues from Rotterdam and Breda, we decided to do a study, a very fast study, to look at the incidence of thrombotic complications in patients admitted to our ICUs, so critically ill patients. And these are the authors of this the study, and you recognize perhaps Professor Huisman, but also Milou Stals, Dr. Van Pasen, Fleur Kaptein, PhD student, and Dr. Arbus here from the LUMC in Leiden. So what we did, actually very straightforward, we wanted to investigate the incidence of arterial or venous thrombotic complications in critically ill COVID-19 patients in the 3 Dutch ICUs. And within a few weeks' time, we had 184 patients with proven COVID-19 pneumonia, all received standard routine thromboprophylaxis, and even in the course of the study, a little bit more than standard thromboprophylaxis. Very importantly, diagnostic tests for thrombotic complications were only performed in case there was a suspicion on any complication. There were no screening tests performed. And at-risk period was from the moment they were admitted to the ICU until they died, until they were discharged from the ICU to the ward, or upon censoring at April 22nd. And these patients had an average age of 63 years old. The majority was male, which is not surprisingly also what Anna just shown us. Almost 4 in 10 had signs of clotting activation, coagulopathy, defined as spontaneous prolongation of either PT or APTT, which is probably a little bit high compared to other cohorts, showing that there were indeed probably in Leiden and Erasmus, especially more critically ill patients than probably in other ICUs. It's important to realize with the results I will show you. We had a median follow-up of 2 weeks, and within those 2 weeks, 22% died, 43% were discharged, and 35% were still on the ICU. And this is very short the result what we found. So an enormous high incidence of any thrombotic complication. So corrected for competing risk of death, the incidence was 49%. And if you do not correct this, it was even 60% as shown in the graph. And this is much, much higher than we expected, but also what we have seen in other, let's say, infectious diseases or any other condition actually for patients admitted to the ICU. And this paper had a major impact because over the weekend, it was Easter weekend, over the weekend, in at least 5 European and also North American countries, guidelines were updated because of the study to increase the dose of thromboprophylaxis on ICUs and to have strict thromboprophylaxis in all hospitalized patients with COVID-19. And also, and this paper had quite some attention from the media and had a good impact on social media, showing that only a small study in 3 hospitals can have a global meaning and change the course of events. We were not the only one doing the studies. Very shortly after our publication, there were many other publications, initially small cohorts. Now there are cohorts of thousands of patients showing the same, a very high incidence of thrombotic complications, both on the ward as well as on the ICU. Interestingly, the range in which these incidences are reported are quite broad, so 17 to 70% on ICUs. And this is caused by different factors. For instance, let's say the patient characteristics. I've already shown you that we had really critically ill patients with a lot of clotting activation. This was different in other studies. Also, we did not perform any screening. And studies in which patients were screened for deep vein thrombosis, for instance, they found 70% in patients at the ICU, and then it's not an incidence, but it's a prevalence, obviously, but still very high rates. Well, with this knowledge, there are a few $1 million questions to answer. How can we best prevent COVID-19? thrombotic complications? How can we best diagnose these complications? How can we best treat those complications, and what's the prognosis of these complications? And we are very lucky to be part of the so-called Dutch COVID and Thrombosis Coalition, which is a coalition supported by Zon en Mee in the Dutch Thrombosis Association, who allows us to collaborate with all academic hospitals and many non-academic teaching and non-teaching hospitals across the Netherlands to answer these important questions. And this consortium has 5 work packages from, let's say, basic understanding of why the clotting activation is so severe, until the interaction between the virus and the clotting factors, until, let's say, more epidemiological studies on the prediction of clotting complications, the prevention, the treatment, and the long-term complications. And I will show you a few publications and studies that are currently going on. So this is from the first work package about to understand why the clotting activation is so severe. And there are several experiments planned within the Beat COVID consortium, so we are very grateful for this collaboration. It's mostly performed from our side by Professor Eikenboom, Kannegieter, and Versteeg. And they will look at the association of changes in clotting factors and clotting factor levels and the occurrence of thrombotic complications in patients with COVID-19, but also to associations of plasma proteome components in the course of COVID-19. Another study by our department in collaboration with Professor Kannegieter, because initially there were discussions whether we were not overestimating the rate of thrombotic complications, and we do see thrombotic complications in other patients at the ICU. What are you talking about? So we decided to compare the rate of thrombosis in COVID-19 patients to those with influenza pneumonia, also admitted to hospitals in the years before the COVID-19 pandemic. It was the work of Milou Stals, mostly our PhD student, and you can appreciate from this slide that especially on the ICU, the rate of thrombotic complications, venous thrombotic complications, is much, much higher in those patients admitted with COVID-19. Interestingly, the other way around, there were more arterial events, so strokes and myocardial infarctions in patients admitted on the wards and ICUs with influenza. That's probably due to the fact that influenza patients are mostly older than COVID-19 patients, which probably they have also more advanced atherosclerotic situations, which may cause this higher incidence. But for thrombotic complications in the venous system, I think this study shows very clearly that we do see a higher incidence in COVID-19 patients. Another very important study recently published by Fleur Kapteijn's first author, also a PhD student of our department. Between February last year and today, also shown by Anna, the treatment of patients with COVID-19 has radically changed. Now we treat them with dexamethasone, until recently also with remdesivir, which of course was not the case 1 year ago. And we had the hypothesis that perhaps because we treated the patients better, their prognosis would be better and the rate of thrombotic complications would be lower. And we compared the incidence of thrombotic complications to patients from Dutch hospitals in the first wave to those in the second wave. And to our surprise, or what we expected actually, was that the mortality was much lower in the second wave. So we are doing something good here. So the adjusted hazard ratio was 0.53, so almost a 50% reduction in mortality. But the incidence of thrombotic complications was the same. So a hazard ratio of 0.86, so perhaps a little bit lower in the second wave, but still it was the same among more than 1,000 patients across several Dutch hospitals. And the reason for this is probably because we have a lower threshold to looking for thrombosis. So we make more CT scans, and by making more CT scans, we will find more thrombosis. But also, we treat all these patients with corticosteroids, which have an intrinsic tendency to cause clotting. So perhaps we also induce new clots by our novel treatments that may save lives, but do come at the cost of clotting complications. And this underlines the relevance of the large ongoing randomized trials worldwide to look for the optimal dose of thromboprophylaxis in patients with COVID-19 admitted to hospitals. Shortly, there was a study performed by Emin van der Wal and from the intensive care unit, and Professor Evert de Jonge, and they looked to the effect of heparin on patients with a proven pulmonary embolism. Because I did not share you this before, there's also a discussion whether this is conventional thromboembolism, so clot embolizing from the pelvic veins or the lower leg to the pulmonary arteries, but it could also be some kind of in situ immunothrombosis. And we are not really sure whether heparin, the routine treatment for clots, would also be effective to these immune thrombotic events. And we looked at all patients at the ICU who received unvaccinated heparin because of pulmonary embolism. We looked to the clotting activation in the course of the treatment with the D-dimer level as shown in the graphs here, and you can see a sharp decrease in D-dimer level after initiation of heparin treatment, probably what we would have expected, showing that there's, let's say, a good response to the treatment. And also these patients received follow-up imaging, and almost all clots fully resolved even within days after the initiation of treatment. So this study showed really that the routine treatment with anticoagulation of these clots would indeed be the good way to go. Also very relevant for our treatment actually, and for how we manage these patients in clinical practice. Very shortly, and what we're planning now in collaboration with all university hospitals in the Netherlands is to take a look at the long-term impact of clots. So we will compare patients with a thrombotic events who survived COVID-19 to those who did not develop thrombotic events, and we will look for functional recovery, quality of life, and the incidence of long-term complications because of non-resolved clots. And we will of course look to whether the clots resolve, and we already saw a hint of this in our initial study, and we will compare this to what we know how clots resolve in the non-COVID-19 situation. And with this we further hope to improve our knowledge, and with that our management of these patients with COVID-19 associated thrombosis. This is my last slide showing all LUMC members of the Dutch COVID and Thrombosis Coalition, and I'm extremely grateful to all of them for the great collaboration, and we will be busy for the next years, I think, to answer all our study questions and hopefully make more, let's say, publish more studies which will positively impact the way how we treat patients with COVID-19. And with that, I'm very happy to answer any questions after the other speakers, so please put in your questions in the Q&A, and I will be back to answer those. Thank you.
Okay, so for me it's a great honor and pleasure to continue presenting on behalf of the immunology group of people that has been involved in the Beat COVID project. And it's really on behalf of a large group of people that I'm trying to present you what we've been learning about the immune responses to COVID-19 infection. Because the SARS-CoV-2 virus is a completely new virus that none of us has experienced before. And the number of people that have seen very similar coronaviruses is also rather limited. So we do have a completely new virus giving our immune system a completely new experience, which is rather interesting from a lot of different aspects. So we are currently assessing not only the local responses, but also the systemic responses in response to SARS-CoV-2 infection. And we can discriminate responses that are quite common, which we may also see in other viral infections, and compare those to the antigen-specific responses that are maybe unique to SARS-CoV-2 infection. And within the Beat COVID group, we are assessing a lot of these different aspects of the immune response to COVID-19. So we're assessing epithelial responses to viral infection. We're profiling what's going on at the mucosal surfaces in patients. We look at inflammatory cytokine and chemokine profiles. We assess the role of complement inhibitors, which may nicely link back also to the coagulation problems that many of those patients do experience. There's detailed profiling of different immune populations in the circulation, which may be again compared to the mucosal profiles. There's a stimulation assays ongoing, but we also assess the very specific responses in terms of neutralizing antibodies, the modifications those antibodies may have, as well as the T cells that may also be induced by the viral infection. And finally, we also try to assess the antigen-specific responses at the site where the infection is actually ongoing. And as the Beat COVID group, we think that the integration of all those different responses is really essential in order to combat COVID. In our ideal world, we are doing a nice study at the LUMC, in which we included a large majority of COVID patients that have been admitted to LUMC. Anna already said we have 200 patients included at the moment, and they were included both at the ICU as well as at the regular wards. We have longitudinal follow-up of all our individual patients, having samples 3 times a week during the entire period of admission. We do all measurements on all samples, and that's possible because the clinical chemistry department is playing a key role in distributing samples from all the individual patients to all the different research groups involved. So we are measuring what is really going on in the same patients. And we have paid a lot of efforts to create a data warehouse in which all data can be aligned for all different patients. And there's also inclusion of all clinical data coming from the clinical wards and the ICU registration systems to align clinical events to the laboratory data that has been identified by the different research groups. We have paid a lot of attention in characterizing and making classifications of the different patients, and we try to make sure that all research groups use the same type of classification and severity scores for the patients over the course of disease. And of course, we have involved people with a great expertise in longitudinal data modeling and data analysis. So it's really a group working together. So it's the first stage in the infection. People are assessing the response of the epithelium, and in this case, the tracheal as well as the bronchial epithelium in response to viral infection. And it has been shown that the further differentiated the epithelium is, the more virus infects those epithelial cells. So there's a true activation of epithelial cells in response to viral infection, and they're currently profiling the different responses that those epithelial cells may have to the viral infection. And within the Beat COVID consortium, also epithelial cells have been collected from the actual patients at the ICU, and those will be assessed in similar systems. So we have an epithelial model to study the response to SARS-CoV-2 infection, which will give interesting insights in the initial activation of defense systems. However, the epithelium is not acting on its own. There's also a lot of different immune populations migrating into the epithelial layers, and especially the nasal mucosa is an interesting site to assess the different populations that may be migrating towards the infection. And indeed, detailed analysis by mass cytometry of nasal mucosal scrapes has identified 28 different subpopulations of immune cells that are present within the nasal mucosa, comprising myeloid cells, but also CD4, CD8 T cells, and several innate immune players. And if we plot those data against the time since the onset of symptoms, being a marker of the disease duration, indeed, several monocyte populations, as well as granulocytes, maybe to a lesser extent T cells, are strongly increased in the nasal mucosa at the most severe stages of disease and normalize when the patient is recovering. So looking at those populations in the nasal mucosa already gives some insights in the players that are involved in disease clearance. Similarly, also in whole blood, these type of cells can be profiled. And there are some very extensive panels that allow a very high level of phenotyping of the different immune cell subsets, including all the different stages of B cells, but also many different T cell subsets, myeloid cells that can be monitored over time in those patients. And currently, data are being aligned over the course of disease and trying to plot it against the different viral loads. But interesting enough, especially the frequency of plasma cells in the different stages seems to correlate with the disease clearance. So it's a very high level of immune profiling that is being performed on all those patients. However, those cells do not get to the circulation independently. They need to be recruited. They need some inflammatory mediators, probably secreted by the epithelial cells, to be recruited to the epithelium or to be developed into the circulation. And one of the factors that we will be assessing in the circulation of those patients are the different complement inhibitors. Because we think that especially complement inhibitors like C4 binding protein or C1 inhibitor are important regulators that provide crosstalk not only between the immune system and the cells being recruited, but also to the coagulation system and probably several others as well. In addition to complement proteins, there's many other soluble molecules that are changing in the serum of those patients. And if we do assess a large panel of more than 100 unique inflammatory mediators in the serum of those patients and look at which chemokines or cytokines may be different over the time course, we do see an enormous amount of cytokines that is different. And if we compare the different disease stages, for example, fatal disease to moderate or mild disease, or severe to moderate disease, we do see that many different cytokines are actually differentially upregulated in the serum of those individuals, indicating that there's a strong inflammatory profile. It was already mentioned several times that IL-6 is one of the parameters that is really involved in the disease progression. And indeed, if we compare patients with moderate disease to severe or fatal disease, we do see an increasing accumulation of IL-6 in the serum. It is decreasing again if patients do recover. So looking at those cytokine profiles in conjunction with disease severity, viral loads, is really informative in those processes that are associated with the disease process. Well, these were all parameters that are quite general and can be found in any disease process. But if we combine that with the antigen-specific responses, there's also indications that the antigen-specific responses are altered by the strong inflammatory profiles. And in this case, immunoglobulins were assessed and looked specifically at the glycosylation of the different immunoglobulins as a marker of their anti or pro-inflammatory capacity. And in this case, SARS-CoV-2 specific antibodies were compared to the total immunoglobulins, and there was a decreased level of specific sugar moieties called fucose groups on the specific antibodies, which indicates that SARS-CoV-2 patients have a very strong pro-inflammatory antibody profile, activating strong responsive cellular subsets. So if you would compare that over the course of disease, and this is again plotted against the symptom onset, looking at the different capacities of the anti-SARS-CoV-2 specific antibodies, there's a very strong pro-inflammatory profile in the early days after symptom onset, suggesting that there is a very strong inflammation ongoing. And those antibodies can be correlated to cytokines, and most of them indeed correlate with a strong pro-inflammatory cytokine profile. But it's really functional differences that are being found. Patients with SARS-CoV-2 infection do not only produce antibodies against the virus, but they also produce T cells that were identified here by looking at the cytokine production of CD4 or CD8 T cells in response to stimulation with different peptide pools of virus components. And as you can see here, patients that suffer from COVID-19 do produce cytokines in response to the various components of the virus. There may be some differences in the type of proteins recognized by CD4 and CD8 T cells, but if we compare all different patients, either CD4 or CD8 T cells can be detected in any individual patients, indicating that it's very common to have also a T cell response against those antigens. So if we would now combine all the knowledge that we have gathered for a single patient, this is just an example of a rather randomly selected patient from the Beat COVID study, which has been on our ICU for quite a considerable time, resulting in a large collection of samples to be analyzed by the different laboratories. And if we assess the viral load in this panel, we do see that there is quite some virus present up until day 2022 of the disease course from this patient, and from day 29 onwards, the patients had undetectable levels of virus. In blue, you see the severity scores, which were calculated based on many different clinical parameters. And a severity score of about 10 is quite a severely ill patient, probably being on the ICU and ventilated, but at a certain moment, he's clearly improving and recovering completely until discharge from the hospital. If we now plot all the different parameters that we have assessed according to the same timeline and compare them to either viral load or disease severity, we do see some very interesting patterns. First of all, if we look at the detailed immunophenotyping of the blood, we do see that there's considerable changes in the composition of the blood at about day 29, which is when the viral clearance starts to occur. We do see that in the pattern of just the total populations of B cells and T cells, but we also do see that by that time, antigen specific, so SARS-CoV-2 specific antibodies start to evolve. We do see that some of the cytokine patterns change even ahead of the improvement of severity scores. Some cytokines are already clearing earlier and some are slightly later. We do see changes in the nasal mucosa for different subsets, although we only had relatively late time points for these patients. We do see strong changes in the antigen-specific IgG fucosylation, suggesting that there's a very strong pro-inflammatory profile normalizing into a more anti-inflammatory profile. There seems to be a strong association with the complement binding protein C4BP and viral load. We do see the involvement of antigen-specific T cells, which come up just after viral clearance starts to occur. And we do see that the responses to toll-like receptor signaling are also quite different in this patient during the different stages of disease. But just by looking in very large level of detail into a single patient can tell us a lot on which processes are ongoing. And integration of all those data responses is the strong point of the Beat COVID group. So just to sum up, we have already gathered an impressive amount of data covering both the local and systemic immune responses, both to the viral infection in general, as well as specific for SARS-CoV-2. We do see that the hospitalized patients have incredible changes to their immune compartments and are quite long lasting. Some of them are even bigger than we've ever seen before. We need to integrate the data, combine the measurements, but that will allow us to obtain unique insights into the response of individual patients and also groups of patients, depending on their disease course or the treatment they have received. And we can, of course, extend this data set to include also the coagulation work or the metabolic profiles that have been determined also on the same patients. At present, data integration is complex. We should include pathway assessments and unbiased hypothesis generation, but I'm convinced that this will help us to beat COVID altogether. So, of course, there's an enormous amount of people involved in this work. This is the same list as Anna just showed you from all the different departments that are involved in the Beat COVID group, complemented with the people from the COVID-19 group that are also responsible for gathering all the clinical data. But of course, these are not the only people that work on the project. It would not have been possible to run all this integrated work with all the technicians, nurses, assistants, students, flow cytometry core, all the other people processing and gathering the data. And I think it's really a very nice example of an integrated group. And with that, I would like to give the floor to Marjolein.
Thank you. The pointer. Right, so I will tell you a little of what has been going on in the molecular virology group that is part of the medical microbiology department at LUMC. And this is kind of a list of the people that are there at the moment in totally random order, I must emphasize. Later on in my presentation, I will emphasize and elaborate a little on what we've done in a project together with Janssen. And so I would like to emphasize these people in particular, particularly Tim Dalebout, Jesse Torres, and Sebe Mieni, who have done a lot of that work for that project. Okay, so as you may or may not know, our group actually has studied coronaviruses for a long time, and even before 2002, when SARS coronavirus broke out, the group already was studying viruses of this order of viruses, so the nidovirus order, of which coronaviruses are one family, and for example, arterioviruses are another. So we have already gained a lot of information and a lot of expertise and experience with studying these viruses. And indeed, we started looking at SARS coronavirus 2 when it broke out in 2002, but also when MERS coronavirus broke out in Saudi Arabia, we started investigating that virus. And we've always thought that that was important to do, although for us here in the hospital, it was also always kind of difficult to argue why we would study these viruses, because we never had patient cohorts or anything to look at. And so the interaction with the clinic was always a point of discussion. But of course, lately that has changed majorly now that we have another coronavirus breaking out. And at the beginning, when we started looking at that in the beginning of 2020 or end of 2019, we frankly thought as coronavirologists, okay, this is going to be something that will look like the SARS or the MERS outbreaks. But of course, this was very different, turned out to be very different. But because we had been investigating these viruses for a while, we were ready to go to also start studying the SARS coronavirus 2 virus. We had basal 3 cell culture facilities in which we could work with live viruses like this at basal 3 levels. And recently we also implemented a facility for basal 3 animal infections at basal 3 level. So in that respect, we were ready to go. But on the other hand, getting the virus here in LUMC was another story. I mean, this took some time because at the beginning of 2020, when we decided, okay, this is a new coronavirus, we should start working on that, getting that virus into the LUMC was still a huge problem. Of course, that became less difficult later on when also in the Netherlands there were cases. But we started with an isolate that actually came from Melbourne, Australia, where a couple of researchers were so kind as to have an isolate, grow that, and were willing to share that with the rest of the world. And so we received that from them. We also actually tried to get virus from Wuhan, but that was much more difficult. So that never worked out. But the Melbourne virus was the first isolate that we started working with in our basal 3 facilities. Later on, of course, as said, there was a lot of other viruses that also in LUMC could be isolated. And right now we're working with several of the isolates that we acquired from the hospital itself. So how does that go? How did we start with that? How do you do that? Well, it's quite simple, of course. You start with some patient material. So usually this is nasal fluid. You put that on susceptible cells, and we knew from our research that vero cells were very likely to be able to support the infection with SARS coronavirus 2, incubate that, and then hope that that virus starts growing in those cells. You can check that by looking at the cells and see if they would die or another form of cytopathologic effect, which would be an indication that indeed that virus is growing. You harvest that medium and then you inoculate larger bottles to expand that virus so that you have a larger stock that you can then start using. After that, you can perform growth curve to see how does this virus grow in the cells that you have chosen. You can analyze the sequence because, of course, as you all now know, RNA viruses as SARS coronavirus 2 are notorious for evolving. So you want to know what's going on in the system that you're using for analyzing that, so the cells. Immunofluorescence assays to look, so what does look at all the viral proteins inside the cell, Western blots, etc., etc. And doing all that is also part of building that toolbox for studying that virus, and that is what we have started doing at the moment. You need antibodies, and we were lucky that this SARS-2 virus actually looks like SARS-1 virus a lot, or the SARS virus that broke out in China in 2002. And so many of the antibodies that we already had against that SARS coronavirus actually cross-react to SARS coronavirus 2, which made it easy to start on that. Well, finding suitable cells for different studies, it's still a struggle that we're dealing with, and in the meantime, we also acquired several different animal models for studying SARS coronavirus 2. I will not elaborate on that. I did that in another top research presentation of some time ago. And what we also did is starting to develop reverse genetics. And this is something that gives possibilities to really look into the genetics of a virus. In this case, what you do, you have an RNA virus. This has an RNA genome. You make a DNA copy of that, and you can clone that in a vector or a plasmid. And then you can manipulate that by making mutations. And these mutations will then teach you about the features of that virus, and it's a very strong way of looking at those viruses. And of course, it was important for us and relatively very easy for us, because we already had this experience, to get funding and participate in many different projects with many different collaborators on this SARS coronavirus virus. These are some of the very first data that we my colleagues produced, looking at the growth curve, so the growth features of SARS coronavirus 2 in these vero cells. And there you can see that in comparison with the SARS coronavirus from 2002, the SARS coronavirus 2 in these cells seems to grow to a little lower titers, but the growth curve looks the same. The plaques look very similar. And this is just to illustrate what we did in the beginning. What was interesting also, that if you start growing this virus in cell culture, you have to think about, okay, how does it behave? And then very soon we realized that something was going on in these vero cells. And the bottom line of that is that vero cells, well, the virus uses a receptor called ACE2, but it also uses a cofactor called TMPRSS2. And vero cells turn out not to have this cofactor. And the result of that is that the virus evolves to lose a sequence here, a recognition sequence for a protease that normally cleaves the spike. This is the spike of the virus that is not on the surface of the cells or of the virus particle. And normally furin is a protease, a cellular protein, cleaves the region between the two parts of the spike. And this is beneficial normally in a human infection to start the infection. But in vero cells, actually, because these cells do not have TMPRSS, this is not of any importance. And the virus will then evolve to lose that furin cleavage site. And that is something that we noticed very soon after starting growing that virus in vero cells. And this is something that we then further investigated. So in due time, we became participants in a lot of different projects around this virus, and we also managed to raise a lot of funding to expand our BSL-3 facility, and we were able to hire quite a few new people so that we could start working on this virus. Now, these three projects, SCORE, CARE, and also PanCoronet, are projects that focus on antiviral agents, and this is something that we still think is very important in the battle against this virus, besides vaccines. And of course, we heard about the Beat COVID project, and Meta later on will talk about this project. But we also embarked on several projects dealing with the development of a vaccine against SARS-CoV-2. And most prominently, I guess, we were involved in the development and testing of the Janssen vaccine here in LUMC. But we also are still involved in other vaccine projects that are also developing it. And well, considering the Janssen vaccine, of course, you probably have heard that in the meantime now has been approved by EMA for use in humans, which is of course a very nice thing, and it's a very, well, honorable thing for us as a virology group to have contributed to that. And well, what exactly did we actually do in that project? Mainly what we did, oh no, first I'll explain what does the Janssen vaccine actually look like? What is the basic design of that? Well, it's a vaccine that is based on an adenovirus. So an adenovirus is a common cold virus, and this is a non-pathogenic virus, and it's not dangerous for people, and it can be used as a carrier of in a vaccine platform, like which has been developed by Janssen a long time ago already. And the trick is to add the code for the spike protein of SARS-CoV-2 in the genetic material of that adenovirus, and in that way that spike protein of SARS-CoV-2 will be expressed during the exposure of that virus particle after vaccination. Janssen, in a very early stage, already realized that they would probably not be the first ones to be ready with a vaccine. And so they figured very early on what we should do is really think very well on how to design our vaccine so that it will be as efficient as possible, and so result in an immune response that is most efficient, so that we might be able to develop a vaccine that can do with one shot. And as you know, this actually, they succeeded in that. And this is from one of the first papers that we already also were co-authors on, in which they tested several different designs of their vaccine, in the sense of that sequence encoding the spike of SARS-CoV-2. And what they aimed at is producing a viral spike that is as stable as possible and in a conformation that is pre-fusion. So meaning that the immune system will have a chance to attack that spike protein before it enters cells, so that you can dismantle the virus infection very efficiently. And so in the end, they ended up with a construct that is, the construct that actually won in the end over all of the other ones, which contains the natural signal sequence here of that gene, which contains that furin cleavage site mutation in order to prevent that cleavage between S1 and S2. And that in this case means that the virus spike is more stable than, more stable, and this is beneficial. And that there's another trick here. There's a few prolines added here, and this is also meant to stabilize that spike. So after, well, in the process of choosing one of these candidates, or choosing in the end this candidate to be the best, several animal experiments were done by vaccinating these animals and then looking at the responses and to see whether those responses would be promising and would tell us which ones of these constructs would be the best. And the part that we did in LUMC here is mainly looking at the virus neutralizing antibodies that were produced upon testing those vaccines in small animals and also in non-human primates. What are neutralizing antibodies? Of course, well, most of you know that, of course, but so part of the antibodies that will be produced by B cells after exposure to the virus, or in this case, exposure to the vaccine, is that these antibodies, if they fit well onto the spike of the virus particle, they can prevent the infection of the virus particle into new cells. And this is one of the branches of the immune response that is regarded very important. So a vaccine should elicit these kind of neutralizing antibodies. And this is one of the branches and one of the important things that you check while designing these vaccines. And this is what we did. Subsequently, of course, they did challenge experiments to analyze protection from infection and to see if indeed those predictions were indeed true. And this is just to illustrate what these kind of analysis or assays look like. And this is something that particularly Seba Miani in my group has been developing and optimizing for our lab, called a microneutralization assay. And the principle is very simple. What we do is we get serum from animals that have been treated with the vaccine. And if these animals produced neutralizing antibodies, they will be in the serum. And so what we do is we mix the serum in different dilutions with a certain amount or certain number of virus particles. And then if there's neutralizing antibodies in that serum, they will stick to those virus particles. And then this mixture of virus particles with that serum or with the antibodies here will then be incubated on cells that normally are able to be infected. So for example, here you have a virus control, meaning that all the blue that you see here are the cells that are still alive and are not killed by the virus. But if you put virus on these cells will be killed, and so your wells will be empty. If you now do that and then add these mixtures of virus with antibody, and you see that the virus is not able to infect your cells anymore, and so these cells will stay blue, then you have high titers of neutralizing antibodies. And this sample, for example, indicates that the serum that is used for this assay has very high neutralizing antibodies, and here the neutralizing antibody is less. So this is basically what we do. Yeah, I will speed up now. Okay, so basically in the end, we contributed to making that choice for that ideal construct, and this was developed further, and of course this was also tested in humans later on, and this is an illustration of that. And there it is important to emphasize that we as a LUMC were also involved in those human trials, particularly in the phase 2, and both CHDR and LUMC have contributed there. So this was a project led by Ingrid de Visser-Kamerling together with Meta Rustenberg, where they did this randomized double-blind placebo-controlled phase 2A trial, as it is called, to test several different doses of the Janssen vaccines and also different regimes, so not the 1 doses but 2 doses in these possibilities. So this was very important and emphasizes that in several ways we have contributed to the development of these vaccines. Of course, after that, The phase 3 was conducted, and as you all know, this resulted in good results, showing that the vaccine is both safe and also efficient, and therefore, in the end, it could be approved by both FDA and EMA for use in humans. So that's a nice success story, but is that finished now? Well, probably not, as you all probably also realize in the meantime, because viruses evolve, and the viruses like SARS coronavirus evolve a lot, and we have seen already some different variants popping up in the population, and every time the question is, do the vaccines that now have been developed still work against these variants? And so we will probably keep working on this for a while now. Is the crisis almost over? I am convinced that the crisis is almost over, but the work and the research on these kind of viruses is definitely not over, and we will, of course, continue with that from now on in the coming years. Thank you very much.
So thank you for having me and having me present on behalf of the team Chico, which is the nickname for the team that has been working on a controlled human infection model for SARS-CoV-2 coronavirus. And I think our previous speakers have made very clear that it's not just a viral infection. There's a very important host response that will determine whether there will be a severe disease and hospitalization, or whether there'll be an effective immune response and clearance of the virus. So this also means that there's considerable heterogeneity in responses of individual to the virus, with varying results, either going into the ICU and pulmonary support, upper respiratory tract infection, but also asymptomatic infection occurring. There's heterogeneity in the mode of transmission. We know that the primary mode of transmission is droplets, but we also know about contaminated surfaces, and there's a lot of discussion about aerosols and the roles of aerosols in transmission. And then there's heterogeneity in host spreading. We know that the R0, if we wouldn't do anything, is somewhere between 2 and 3, but we also know that for any infectious disease, and this is also true for SARS-CoV-2, there's a small part of the population that's responsible for the biggest part of the transmission. So there's a 20-80 rule for SARS-CoV-2 virus. 20% of the people are responsible for 80% of the transmission. And on top of that, we have heterogeneity in the virus, and this is something that Marjolein already alluded to. So here I'm going out of my comfort zone, not being a virologist, so I have to pay respect to Mariet Veldkamp and Eric Snyder, who taught me on the topic. But the virus is continuously evolving and mutating at different sites across its genome. And if you read the news, you think it only happens in the spike protein, because that's what's on the news all the time, because the spike is obviously the thing that we're interested in, because that's what our vaccines are based on. And we're typically afraid that the virus will escape the coverage from our vaccines. And again, this is something that we don't know. And so we are now talking about the United Kingdom variant, the South African variant, and the Brazilian variant, but I've been taught that this is an oversimplification of the dynamics within the virus, and also a lot of unknown changes that occur in regions other than the spike protein. But we do know that there's a geographic difference between the spread of these different types of virus, which obviously also will change as time will continue. So we have very important questions that we still need to address for COVID-19 and that still haven't been addressed. One is the heterogeneity in clinical presentation. So who will go on to become symptomatic and who will present within a hospital and who will not? And obviously there's risk factors, but on an individual level, we can really not predict this. There's heterogeneity in infectivity. So who's actually responsible for transmitting to many of these people? And again, that's something that we don't know and we cannot pinpoint. And then there's heterogeneity in immune responses. So there's people that apparently develop very efficient neutralizing antibodies or CD8 responses. And then another complicating factor is the variant of concern. So to what virus are they, developing these kind of responses, and to what virus are they not protected? And to complicate things even more, there's also a factor time. So the question really is, at what time of exposure do these things occur, and are they somehow related? And most likely, obviously, they are. So there's several ways to actually try and answer these questions, and obviously Marjolein already alluded to that as well. There's the development of lots of different animal models. However, there's a clear heterogeneity in animal models, as COVID-19 is an immune-mediated disease. So depending on the immune system of the host and host animal, you might get different outcomes. So even within non-human primate models, one non-human primate model might be completely asymptomatic, whereas another model may get pulmonary infections as humans do. So there's differences between models. On the other hand, you can do epidemiological studies. You can study our patient population, as we're doing obviously in our Beat COVID program. You find you can capture the full heterogeneity of life, which is, I think, is a main advantage, but obviously you don't have any data from before people were infected. There's a large number of asymptomatic infections which are very hard to study because particularly people don't notice that they're asymptomatically infected, and there's usually a limited level of detail, although that's something that we've been trying to really overcome in the Beat COVID program. So right in the middle of these two sit controlled human infection studies, and I just want to emphasize this because it means that controlled human infections by nature are a way to sort of close the gap between animal models and epidemiological or real life studies, and so they are a model in itself. They will aim to include a very homogeneous group of people, typically adults, young, healthy adults, and they try to balance homogeneity with heterogeneity. So getting a more homogeneous group of people with a homogeneous dose, with a homogeneous virus, to really understand the complexity of the issues that are ongoing. And obviously that also means that there are limitations to controlled human infection studies by definition, because a model is still a model. Controlled human infections have been proven to be very useful in history, although there's also quite some dodgy history to controlled infections, which we definitely should not repeat. And then Koch's postulate has been cited much, and I have a couple of examples here of things that we now take for granted. So transmission of yellow fever by mosquitoes, transmission of dengue by mosquito, different dengue serotypes, even dengue vaccines. All of these things wouldn't have been developed without controlled human infections, and the difference between neutralizing stomach acid and then getting infected by salmonella with and without neutralization of the stomach acid is obviously something that was also discovered with controlled human infections. So in the past they have proven to be extremely useful, which is why now we are increasingly using these infection models to study different types of infectious diseases. And within the LUMC, I've learned we even have a history of controlled human infections doing rhinovirus infections for quite a long period of time. There's a wide array of pathogens that are now being used to purposefully expose people to, and I will not go into the details of all these different models, but you can clearly see that particularly for influenza and malaria, there's really an increasing interest in using these type of models, also for the development of vaccines and drugs. So within the LUMC, we have a unique expertise center whereby we have different controlled human infection models that are continuously ongoing. And this is really unique. There's no other center in the world where we do multiple pathogen exposures. We do hookworm infection studies, we perform schistosomiasis infection studies, and we purposefully infect people with malaria. And these studies are ongoing all at the same time. And again, that is something that is really unique on a worldwide level. And that has led to very interesting results and discoveries. And this is just some data that I borrowed from San and Maria, who've together published an extremely nice paper about the differences in immune responses to malaria between naive European and semi-immune African population, digging into the details of the different T cells that are present in peripheral blood and showing how different subsets differ between different populations, really giving us keys and new leads to develop novel vaccines or protective strategies. Similarly, we've done the same for schistosomiasis. It might be a disease that's unknown to many of you, but it's actually a disease of global importance with about 200 million cases infected around the world. And we're studying this disease at a very detailed level by making use of a controlled human infection model where we actually map clinical symptoms, antibody responses, cytokine responses, but also the development of the worm with a very special and highly sensitive antigen detection assay developed by Paul Korstjens and Govert van Dam. And then Simon Jochems put all of that data together and showed that with that data, you can actually predict who becomes symptomatic after schistosome exposure and who does not. And again, despite the fact that it is a global disease that has been around for decades, this was extremely novel and would not have been become known if we wouldn't have developed a controlled human infection model. So the question now is, can we do a similar thing for SARS-CoV-2? And the step one would be to obtain the virus, because obviously you need to obtain the virus and you need to obtain it in a quality that would be fit for human use. And again, this is not an easy endeavor because you need to have access to the virus. We obtained a virus from one of our patients of a nasal pharyngeal swab right in the beginning of the pandemic in March when the first patients came in. And we grew this virus thanks to the expertise of Maya Lyons and Eric's group, who already had such a long history of studying coronavirus, had all the techniques ready to actually isolate and culture this virus, and were able to subsequently show in novel ACE2 receptor positive mice that these viruses are actually this virus is actually pathogenic. And then the next challenge is to be able to produce that virus to a level that you can put it into people. And for that, you need a BSL-3 GMP facility. And I can tell you they are extremely rare, but they happen to be on the 10th level of our hospital. So with the help of the team from the IGFL, the IGFL, the GMP facility that we have here, as well as our biosafety officers, Gijsbert, who's indispensable in this process, we were able to actually get two people, you see them here in the picture, Heidi and Jessie, in a GMP BSL-3 environment, which basically means they have to protect themselves from being infected, but also have to protect the virus from getting infected with anything else but the virus. So this has been a very complex process, which we gone through, and I'm very proud to say that we were able to grow this virus on GMP cells, which we obtained from Batavia Biosciences just around the corner. We've characterized it fully by next generation sequencing, and we now have 200 vials of 10 to the 5th PFU in 0.5 ml stored upstairs in our hospital, and we baptized the virus Chico. Because we had the Chico nickname already for the group. And that was all done in a speed that has been unparalleled. So we were one of the first to be able to do that, and got calls from people around the world asking us whether we could be a backup for all kinds of other projects that were ongoing. So this has been unique. The second step would then be, well, how do we then go on with establishing the model, and what will we do? How will we administer this to people? And obviously the prime question here is, can we do this in a safe way? And can we also detect the virus as we put it into these people? Can we actually detect viral replication in the upper airway? And just to give you some idea of how that would happen, you basically pipette the virus into the nose of healthy individuals, and then try and see as days progress if you can see viral replication. And then obviously there's all kinds of interesting things that you can do and you can study. And then obviously the next question is, should we, what kind of dose should we use? Well, there's a lot of debate about how much virus do we actually need, because we don't know, we don't have any clue. So you start very low dose, low number of people, and then you see what happens and you try to gradually increase. These are very, very low numbers of people. Again, 3 people to start with, potentially maybe even 1, and then going up to groups of maybe 10 or 20 max. So these are really, really very, very low numbers. Obviously, the first question that comes to mind, can we really do that, and is it safe to do so? There's obviously a risk when you do these kinds of studies. There's a risk of severe disease. We still don't have 100% effective treatment. So we have to take that risk into account. You run a risk of complications. We've just heard Eric talk about thromboembolic complications. You have complications of long COVID, people harboring long-term symptoms. There's risks of transmission beyond the trial center. Obviously, we don't want the virus to go out there, although it's already there. And there's a risk of boredom, and that might seem a bit irrelevant at this time. We believe that this is a very serious risk, because you will have to confine people during the trial period to a trial center, and obviously having them run away would really be problematic. And so we got help from the epidemiology department led by Frits Roosendaal to try and actually grasp these risks and get a hold of these risks and estimate how big they would be. And we looked at countries around us, and for every country we calculated the risk of individuals in a certain age group to be hospitalized, to be admitted to the ICU, and or potentially to die. And this was all done by Vince. Has been a tremendous amount of work, and we were lucky that Frits and his group had collected all the data from right at the beginning of all the people admitted in the Leiden region. And so you can calculate the risk of any individual in this area to be admitted to the hospital. And these are the numbers that we came up with. So there's not a lot of variation whether you live in Leiden or in Germany or in Denmark. The risks are sort of the same. And it means that for any age group below 30, the risk is roughly at around 1 in 1,000 for hospitalization, and obviously for ICU admission and death, the risk is much lower. So yes, there's a risk, and yes, we can quantify it. You can do the same thing for long COVID, obviously, and there's all kinds of risk mitigation strategies which you can take to prevent transmission or to prevent boredom. Does that mean that it's definitely a no-go, that we cannot take this risk? No, I think we take risks all the time. If you want to take risks, you can look up websites where you can participate in all kinds of study. This is a study where you can actually get an induced panic attack and then test a new a drug to see if that reduces panic. But there's all kinds of studies, and any study, medical study you will participate in, carries a certain risk. So carrying risk, doing research and carrying risk is not unique. The question really is, does it weigh up against the social benefits? Another example, we're doing field lab experiments now. So we are experimentally putting people together in a setting where they could potentially transmit. They've been tested all beforehand to try and see how people move and whether transmission occurs in a group setting. And obviously that's also taking a risk in some sense. So taking risks is not unique in clinical research. The question is, does it weigh up against the potential downstream value of having such a model? So what would be the downstream value? Well, obviously you can look at correlates of protection. So you can look at who's protected, who's not protected, and what immune parameters determine that. You can look at longevity of protection. How long are we actually protected for? You can specifically look at vaccine-induced protection, and this is something obviously a lot of people are wondering now that they're being vaccinated. How long do they protect them for? Do they protect them against this variant of concern or that variant of concern? And these are all questions that are really bugging us, and these are things that we could potentially answer using this type of model. So I now come to a conclusion. Controlled human infection studies provides insights into SARS-CoV-2 infection at a level of detail that you cannot do otherwise. And the question really is, there's a cost, there's a risk involved, and should we or should we not take that risk for the benefit of science? In the UK, obviously, they've already made this decision, and they've started by inoculating people with the SARS-CoV-2 virus actually Saturday 1 week ago. So these trials are already ongoing. And with that, I'd like to end and really emphasize that the fact that we have been embarking on this project is completely owed to the expertise that we have here at the LUMC from the virology department, from the GMP department, epidemiology, biosafety, and also obviously all the clinicians that have helped me think along these kind of protocols and the risks that we should or should not be taking. Thank you.
Thank you very much, Meta, and all the speakers for these excellent presentations. Really interesting what's going on here at the LUMC. There were a lot of questions in the Q&A, so I would like to start with Erik Klok. We got a lot of questions for you.
Oh, very good.
I would like to start with some general epidemiological questions. Did you see any gender difference in your risk of thrombotic complications?
That's interesting. It's a little bit difficult to investigate because the majority of patients is male, and also the majority of patients with the most severe presentation of the disease and admitted to the ICUs is male, and those are the ones that actually go on to develop the thrombotic complications. So yes, there's a big disadvantage of the male sex, but not because they're males, or at least not because they have a higher clotting tendency, but they have a higher tendency to getting more severe disease.
Yes.
So I think that's the main explanation for this.
Very clear. And there was one question. I don't know if you mentioned this in your presentation. Do patients on the ICU have a higher risk of thrombotic complications than those on the ward?
Yes, definitely. So it's on the ICU, at least in our case series, it was, let's say, every 1 of 2 patients, so 50%, and on the wards it's more an order of magnitude of 10-15%, including all kinds of thrombotic complications, also strokes, myocardial infarctions, and venous thrombosis.
Yes, and you also...
So there's a big difference. And interestingly, while in the first wave we diagnosed these pulmonary embolisms because it was mostly pulmonary embolism at the ICU, but in the second wave, with a lower threshold of testing, approximately every other patient admitted to the hospital will get a CTPA to look for pulmonary embolism. And we now diagnose the PE before they go to the ICU, but still they end up at the ICU.
Yeah. And there was one question, but I think this has to do with the last question. Why is the range of the thrombotic complications so wide? Why is it between 17 and 70%?
Yes, so it's a good question, but every ICU is different, every hospital is different, the case series, the case mix is different, and also the threshold for testing is different. For instance, when we look to the Italian population at the ICUs in Milan in the first wave, I was told by a good colleague there that they had not sufficient CT scan time to put all their patients to the CT scan because there were so many patients. Any regular ward was turned into an ICU, and they just started anticoagulant treatment where they could not explain the clinical situation of the patients. So, of course, if you do not make a CT scan, you will never diagnose pulmonary embolism. And then you get relatively low rates of pulmonary thrombotic complications. On the other hand, if you do screening studies, so performing an ultrasonography of the legs every other day, you will, of course, find more clots. So that's the high end of the spectrum. And also, very importantly, mostly the studies from the US have lower prevalences of incidences of thrombotic complications, but they tend to give more anticoagulation to their patients. So they have more stringent thromboprophylaxis regimes. So this, of course, also may be different because you may prevent clotting, but also in patients on anticoagulant treatment, we have not a tendency to go to the CT scan and look for pulmonary embolism because we think, well, they're already treated. Yes. So, but this is unfortunately not the case because many of the patients we identified with pulmonary embolism already were on full therapeutic anticoagulation. So it's not, you cannot prevent everything with it. Yeah. So that's why there are so many differences. And it's also interesting to see that there are more than 30 meta-analyses already on this topic, and they all have different estimates. There was a very interesting paper published last year, and it's on the pandemic of redundant meta-analysis, because I think lots of the science that had been done in the past year may be considered redundant because the quality is very low, the results are speculative, and it doesn't really help us to move forward.
Okay, I think that's a very nice conclusion of the questions, the discussion on your presentation. Thank you very much, Erik. I would like to invite Simona for the discussion on your presentation. So there were a few questions on the antibody glycosylation. So do you think this could be of use in a predictive model, for example?
I'm not sure if the glycosylation could be applicable in a predictive model, because it's quite a complex association between functionality, and I think you would rather have to assess the functionality of the antibodies also for activating, for example, ADCC, rather than just looking at the glycosylation profiles in there. And of course, there is a lot of variation between the individual patients, which looks very nice if you group them all together in a large plot. But if you have to determine it at the individual's level, I think it's not sensitive enough.
Okay, and then there was one question on the mechanism of this glycosylation, because you had to show a lot of data. You can go into it very, very deeply. So how would this glycosylation change according to disease severity? Do you have any idea?
Well, the glycosylation is regulated by many different enzymes, and those are all regulated and expressed by a variety of different cells, where also inflammatory cytokines and inflammatory environment, like type 1 interferons or interferon gamma, TNF alpha, may affect the activity of those enzymes. So looking at the glycosylation, maybe a reflection of the inflammatory environment the patient has been in the period before that.
Yeah.
So it could be a reflection of a strong pro-inflammatory profile if you see those changes, especially in the antigen-specific immunoglobulins.
Yeah, so that would fit the results.
That would fit, yeah.
Okay, thank you. And do you know anything of these glycosylation profiles in other viral infections?
Well, I don't know all the details on the viral infections, but I know that in many chronic infections, also, for example, in tuberculosis, you see similar changes, and there's also temporal changes in the glycosylation patterns of those different antibodies. Yeah. So I think combining the antigen-specific antibodies and looking at the glycosylation pattern, you probably will find changes like this also in other acute viral infections, and more in the infections that are more longer lasting, because then there is a longer exposition to the inflammatory environment.
Okay, thank you. And then there was one question on the cytokine results, which is of course really your subject. Have you identified any cytokine or a group of cytokines that we could use for a model, or maybe for immune surveillance? Or this was also a question from someone from the CKCL, the laboratory.
Yeah, we are working on it, because it's of course very difficult, and you can look at the data both in a cross-sectional as well as the longitudinal data, and some of the cytokines do influence each other, some are regulated by each other or produced as a consequence of some of the other cytokines. So we're currently trying to do pathway analysis to identify which ones are the most key regulators, but we have not yet identified them. I think there are several obvious candidates, but of course they will also need to be validated before they can be applied in any clinical decision making.
Well, I think there will still be enough patients to validate.
I'm afraid so.
Yes. Okay, thank you very much, Simone. I would like to ask Marjolein to come forward. So thank you for your very nice talk and the insight in what you're doing in this BSL-3 lab, which is of course a mystery to a lot of persons. And so as a clinician, what I found so striking is the difference in protection that your analysts have, your technicians have on the BSL-3 lab, and what we have as protective measures when we go to the patients.
Yeah.
So how do you explain this?
Yeah, difference? That's a big question. I mean, in the beginning, as said before this broke out, we never had a lot of interaction because we never had patients, of course. Yeah. And this in the beginning, we were confronted with that particular difference. We are so, that's really, so apparently also the rules are different, working with the virus and research circumstances as opposed to the more clinical. So it's just the BSL-3 rules are like this, and this is how we have worked always, but it was striking to us as well that there was rather big difference of how we deal with that. And of course, now that we're working with this virus at BSL-3 level still, because this is still the rule that is BSL-3 level, it's kind of weird for us also, for the people working in those labs, that they're so well protected in the lab while holding the virus there. But going outside on the streets, you have more chance of getting infected than in our lab. And this was, of course, definitely different before.
Or maybe it could have to do with the viral load you're being exposed to.
Yeah, of course, we are working with high titer stocks, and of course a tube with a milliliter of a lot of virus particles is something else than encountering somebody who can infect you, of course. And that is definitely one of the factors.
Okay. So there were a lot of questions on the vaccine and for example the spike protein, and I was wondering how difficult is it to change, for example, the spike protein in the vaccine vector?
Yeah, this is relatively not a big deal. I mean, we have now, well, these kind of vaccines are made with molecular biology technology that is now very common. And so it's a matter of changing the sequence of that adenovirus construct that they are using for making these virus particles that can function as a vaccine. And this is not a big deal. So it's just, of course, choosing which mutations exactly you want incorporated and making sure as much as possible to have a new vaccine that is also useful and efficacious and to different kind of variants out there, and these variants may keep coming, of course, and that is kind of something that we have to pay attention to. And of course, this is something that has been known from influenza a lot. I mean, it's obvious that every year we have to do that. Yeah. I don't expect that this should be necessary for these kind of viruses because it does work differently, but still these are viruses that evolve, and as said, we have seen several variations already that might circumvent some of the response to the older variants that were used in the vaccine.
So do you know if Janssen is already anticipating?
Well, I suspect they're not officially telling me that, but I suspect, and I think other vaccine producers that also have vaccines are already acknowledged that they are working on changing their vaccines to adapt to the variations. Yeah.
Okay, very comforting to know.
Yeah, although I mean, I'd like to emphasize that the vaccine that is now out there certainly still works quite well, not perfectly, but definitely to an extent. And I think partly that is also because of that design, in this case for the Janssen vaccine, that was very good, and which gives a very sturdy and very reliable response.
Yeah, okay, thank you. There's one more question. Would the mutation rate of the virus vary in different cell lines without the strong selection pressure?
Without a strong selection pressure? Yeah, it might definitely be different in different cell lines because there's different factors that can influence in evolution, of course. And in this case, for the vero cells, it was because of that factor missing in vero cells, and then the virus does not need that furin cleavage site anymore, and when a virus doesn't need it, it will automatically evolve to lose it, because any mutation that is made, and every time the virus replicates there is mutations made, the mutations that will eliminate that site will increase the infectivity to this cell line, and this is what we have seen.
To make it more fit.
And make it more fit, and that mutation will then prevail and dominate. And so other circumstances and features of other cell lines may indeed inflict similar kind of directions of evolution, and will change and will benefit certain mutations that will then become dominant. And this is of course what's happening in the real world as well. All kinds of factors. Also now the fact that we're going to start using, that we're already starting using vaccines, is another evolutionary pressure to the virus, and this is something we really have to monitor well.
Okay, thank you very much. Those were the questions. I would like to invite Meta for the last part of the discussion. Thank you very much for your interesting presentation. I was somewhat surprised with your last slide that they started already in the UK, and I saw a picture of someone injecting something, but I guess that's not
what happened. I mean, no, that is definitely the wrong picture, and I think this is. I have to emphasize that there's a lot of this in the media as well, because obviously it catches a lot of attention, and lots of these pictures are not, I think, appropriate, or stories are not appropriate. But yes, they have started with a very low dose of virus. Yeah.
So when do you think we would have a model?
Well, it really depends. So I think the first step, and this is also what I suspect they're doing in the UK, is the dose escalation. So try and look at what dose do you need to get a certain level of viral replication. And it really depends on whether that's a low dose. and then very quickly you reach your endpoint and you can move on to the next questions, or whether actually the dose is pretty high because we're so careful that we're only taking small steps in small groups at a time, and that just takes time. So it needs a bit of patience to get there.
Of course.
Yeah. So so that is really the rate limiting factor now, I think. And at some point, people will all be vaccinated. So that's also a sort of limits, I think, the extent to which we can study naive immunity. In this moment.
That's true. And so in the media you read of people who volunteer for these. Have you already do you have a database?
No, so I don't keep databases of people, but there are a lot of people have emailed me with requests. So I think it's important for people. Them? Well, it's important for people to understand that there's another side to it. So there's people obviously saying this is too risky, this shouldn't be done, which I fully appreciate, because that's obviously I can appreciate that, you know, taking a risk is something that we need to agree upon. But there's also a group of young people who say, well, I'm going out every day, I'm getting infected anyway. Why can't I be allowed to be infected for the purpose of science? And I don't want any people telling me that I can't. Because it is my right to actually be allowed to participate in these kind of trials. And this is where the whole One Day Sooner initiative came from. And there's a whole website. So I usually refer people to that website where they can sign up. And there's a huge group of Dutch people that have, you know, said that they're willing to be infected. And obviously there's another side to it. I mean, there's also an advantage maybe to have had this infection, because it may give you some level of protection. So people also see it as, you know, it's going to happen to me anyway, I'd rather do this for science, and then you know, I end up
more
being
controlled, and then I might have benefit from it. Okay, interesting. And then more about the model. So of course you want to include only young healthy persons. So how representative would that be of the people who are admitted to hospital, which are mostly older?
So it's not. So if you want to know about what happens, I mean, what COVID-19 will do looks like in an 80-year-old, you shouldn't do a controlled human infection study, obviously. There's just limitations to what you can and cannot study. I think the main advantages is the fact that you can, from the start, without anything happening, from baseline, monitor what happens over time, and also allows you to go back and see if there were certain factors in these individuals that ultimately allow you to predict what would happen at a later time point. So it really tells you who's infectious, who's, you know, at risk, who's, and that you can really tease out. But obviously that's appropriate for a young population, and some of these markers may also hold true in an older population, but that obviously you don't know in advance.
Okay. So I'm very curious of when this will happen. Thank you very much. I think we have come to the end of this very interesting top research seminar. So thank you all for joining us, and a very lovely evening to all of you.